Hello students, welcome to another lecture on Understanding Biology chapter 23. Today we are going to learn about protists. So at the end of this lecture, you should be able to describe the earliest evidence of eukaryotes, illustrate how endosymbiosis relates to the evolution of mitochondria, explain origin of chloroplast, explain why mitosis is not believed to have evolved all at once, describe the features that distinguish protists from other eukaryotes. When you think about evolution of eukaryotes, for more than half of the long history of life on Earth, all life was microscopic. For more than 2 billion years, Earth was dominated by prokaryotes. As you all know, you have learned about it earlier. So these prokaryotes lacked internal compartmentalization and membrane-bound organelles. The first evidence of appearance of eukaryotic organisms with internal compartmentalization and membrane-bound structures were found in fossils dating 1.5 billion years. Even though evolutionary events that took place at these early stages of life is not yet very clear, now we have recent DNA analysis that suggests that 5 supergroups of eukaryotic evolution, which is depicted by this tree. Eukarya are grouped into 4 kingdoms here: Protists, Plantae, Fungi, and Animalia. We are going to elaborate on this and learn about different phyla, and then we are actually going to learn about how eukaryotes are evolved. So evolutionary history of eukaryotes, like one of the major events that distinguish them from prokaryote is the presence of a cytoskeleton and having compartmentalization, such as nucleus and organelles. So this internal structure of eukaryote is one of the very prominent feature that separate them from prokaryotes. So there are several events that might have aid in this internal structure of eukaryotes. These might have included horizontal gene transfer, which you learned about in previous lecture, enfolding membranes and engulfing other cells. So when you think about origin of nucleus and endoplasmic reticulum, many prokaryotes have enfoldings of their outer membrane extending into the cytoplasm. The network of this internal membrane is in eukaryotes is called endoplasmic reticulum, or ER. Similarly, nuclear envelope is thought to have evolved from such enfolding in eukaryotes. As you can see in this picture, you have prokaryotic cells and prokaryotic ancestor of eukaryotic cells, where they have this plasma membrane is enfolding into the cytoplasm now. Once it's enfolded, you can see that now endoplasmic reticulum and the nuclear envelope has developed. Now let's look into origin of mitochondria. When we talk about origin of eukaryotes, one of the major things is that they have this mitochondria as a energy house or powerhouse in the cell, which is membrane-bound organelle that provide energy. So we all know that when two organisms live together benefiting each other, this type of relationship is called endosymbiosis. So bacteria living in other cells and performing different specific function for their host cells are called endosymbiotic bacteria. Because of their widespread presence in nature, this led biologist Lynn Margulis in early 1970s to come up with this idea of endosymbiosis theory. In endosymbiosis, this is now a widely accepted concept regarding how this eukaryotic early evolution of eukaryotes have happened because of the origin of mitochondria and chloroplast. As for the origin of mitochondria, the original ancestral host cell was anaerobic with hydrogen dependent metabolism or metabolic pathway. this cell might have engulfed a aerobic free-living bacteria with a respiration form that produce H2. This host cell depended on symbiont for H2 under anaerobic conditions, but later when Earth became more oxygenic, they developed or adapted to oxygen-rich atmosphere later. This is how mitochondria was origined, when the ancestral cell engulfed a free-living bacteria that has this respiration pathway and adapt it into the evolving environment later. So another thing that is important that we have to talk about is secondary endosymbiosis. So there are two different types of endosymbiosis, primary endosymbiosis and secondary endosymbiosis. So the mitochondria and chloroplast are considered to have like primary endosymbiosis, where similar way where the mitochondria evolved or origined, similar way chloroplast also origined. In this scenario, the free-living photosynthetic bacteria or a cyanobacterium was engulfed by the ancestral cell and formed an endosymbiotic relationship. All chloroplasts are likely derived from a single line of cyanobacteria, but the organism that like bear these chloroplasts are not monophyletic. If you look at all the organisms that have chloroplast, they are not monophyletic. That means that this was due to secondary and tertiary endosymbiotic events. Both red and green algae obtained their chloroplast by engulfing free-living cyanobacteria through primary endosymbiosis, while brown algae most likely obtained their chloroplast by engulfing one or more red algae in process called secondary endosymbiosis. So now you know the difference between primary and secondary endosymbiosis. So it is very important for us to look at some of the evidence to support this theory. So let's look at some of the evidence. So present-day mitochondria and chloroplast contain their own DNA, which is very similar to bacteria in size and character. During a long time, mitochondria exist as endosymbiont. Much of their DNA and genes have been transferred to the chromosome of the host cell, but each mitochondria still has its own genome. It's a circular DNA molecule, and this is one of the evidence that they are bacterial origin, that living as symbionts. And another evidence is mitochondria also have their own ribosomes that are much smaller than eukaryotic ribosomes, but they're very similar to bacterial ribosomes in size and structure. And as you know, many antibacterials or antibiotics that inhibit protein synthesis in bacteria also can inhibit the protein synthesis in both mitochondria and chloroplast, suggesting that these are bacterial origin. And both chloroplast and mitochondria replicate in similar to bacteria using this binary fission type of replication. So that is also another evidence of endosymbiosis, that they are replicating similar way. And another thing that is very important to understand is that the mechanism of mitosis and cytokinesis, when cells are dividing, mitosis and cytokinesis happen, we all know it's a common process. It did not evolve all at once. In fungi and some Protist groups, nuclear membrane actually does not dissolve. So this is like, it is actually not all happen at once. So now when you think about eukaryotes, their nuclear membrane actually dissolve when they are getting ready to replicate. So it is believed that mitosis evolved in eukaryotes in stages. Now that we learned about the origin of eukaryotes, let's look at the earliest organism, eukaryotic organism. So Protists are the most ancient eukaryotes that are classified on basis on one single negative characteristic, that they are not fungi, plant or animals was the one negative characteristics that they use to characterize protists. But protists are actually highly variable with no unifying features, so it is very difficult to categorize them into groups. But modern molecular and DNA sequencing data actually help us to categorize and put them into a phylogenetic tree to understand the evolution of protists. But in this lecture, we are actually going to define protists and look at their unifying characters, some of the characters that we need to understand about regarding protists. So there are variable characteristics among protists. These protists can be unicellular, some of them can be colonial, some of them can be multicellular. Most are microscopic, but some are really large, such as brown algae, green algae, and they can actually, when you look at their shape and structure, they can have all different kinds of symmetries, and they can have all types of nutrients. So let's look into several characteristics of these. The first one we are going to look at is locomotion. So protists actually move using flagella. They use flagella to propel themselves through the water, but some other actually use cilia. They actually use these cilia to create water currents. Some amoeba can have pseudopodia. Pseudopodia actually translate to false feet, and they use this pseudopodia for locomotion. And other protists have exopodia, which are thin branching protrusions supported by axial rods. They can be extended or retracted. So we have pseudopodias can be lobopods, philopods, and exopods. So these are different kinds of false feet that protists use for locomotion. We have cell surface structures. Many protists have delicate surfaces that commonly have plasma membrane, and some others have extracellular material, such as diatoms have silica cells that are in different shapes and structure with many different colors. So cell surfaces can be vary depending on what protist that you are looking at. And also protists have the ability to survive harsh habitats despite of their delicate surfaces. This is due to the ability to form tough cysts. A cyst is a dormant form of a cell with a resilient outer covering where the cell metabolism is shut down for a while until that unfavorable condition is actually solved. Then it can regrow, and this is actually used for disease transmission for some of the pathogenic protists that we will look in future. Another characteristic that we are going to look at is nutrition. Protists can be heterotrophic or autotrophic. They can be prototropes, heterotrophs. Prototropes engage particular food matter. So let's look into each category a little bit here. So prototrophic protists obtain energy from organic molecules synthesized by other organisms, while heterotrophic protists are prototopes. They engage food particles, creating vesicles or pagosomes in the protists. These pagosomes will fuse with the lysosome and digest the food within. Some autotrophic protists are photosynthetic, and some others are chemoautotrophics. So these are protists that are both. There are some protists that have both phototrophic and heterotrophic, and these are called mixotrophic protists. So now we are going to look at multicellularity. So as the cell became larger, the surface to volume ratio became smaller. Therefore, multicellularity was evolved to solve this problem. multicellular organisms have many cells that are associated with one another, and this multicellularity actually allows specialization to this distinct type of cells, tissues, and organs, which allow to have a different function. This division of labor within the body allow organisms to process cells devoted to specific function, such as protecting the body and then reproducing, especially. And this might have arisen multiple times, and it is one of the very important features of eukaryotes to having multicellularity. One of the example is volvox. And sometimes these protists can form colonies. A colonial organism is a collection of cells that are permanently associated with each other, but with little to no integration of cell activities. Many protists form colonial assemblies. One example is volvox. In volvox, there is a discretion between colonial and multicellularity, because sometimes it's difficult to determine if it is multicellular or if it is colonial. So the true multicellularity, on the other hand, is where the activities of individual cells are coordinated and the cells themselves are in contact with each other, and this is called true multicellularity. It is one of the major characteristics of eukaryotes. Three groups of protists actually have independently attained true but simple multicellularity, and these include brown, green, and red algae. So one final characteristic we are going to look at today about protists is their reproduction. So it's one of the most important characteristic. And when you think about reproduction, protists actually normally reproduce using asexual reproduction, but some have an obligate sexual reproduction stage, and some undergo sexual reproduction due to stress. So like I mentioned, asexual reproduction is the typical mode. Some species have an unusual mitosis. Mitosis actually give rise to equal sized daughter cells. Budding sometimes give one daughter cell smaller. Sometimes schizogony is actually cell division preceded by several nuclear divisions, produce several individuals. And like I mentioned, sometimes they have sexual reproduction as well. This is called obligate sexual reproduction. And it is one of the features that is very important. So in protists, mitosis, nuclear membrane remain intact through the mitosis, where mitotic spindle form within the nucleus. In other protists, budding can happen, where daughter cell bud from the mother cell and they are smaller. Some other species can split into two equal halves after mitosis. In some protists, cell division proceed multiple rounds before the nuclear division. This allows cytokinase to produce several individuals simultaneously. So these are actually different types of asexual reproduction. Now let's look at sexual reproduction. So sexual reproduction actually occurs through mitosis, which one of the major evolutionary innovations arose in ancestral protists that allow to produce haploid cells from diploid cells. In sexual reproduction, offsprings are produced through fertilization by combining two haploid cells. So one of the major benefits of sexual reproduction is genetic recombination, and most protists undergo sexual reproduction only in time of stress. So that is the end of this lecture. So in summary, eukaryotes are organisms that contain a nucleus and other membrane-bound organelles. Endoplasmic reticulum and the nuclear membrane are believed to have evolved from infoldings of outer membranes. Mitochondria and chloroplast evolved from engulfed bacteria that remained intact. There are several evidence that support endosymbiosis theory, And all protists have plasma membranes, but other cell surface structures and components are highly variable. I hope you enjoyed this lecture, and until we meet from another lecture, have a good day. Thank you.